Hello, my 10th CBSE biology students, and today we are going to start with chapter 8, How do organisms reproduce? So, before going to reproduction, we have to know the definition. So, the process of production of new organisms from the existing organism of the same species is known as reproduction. What is the significance of reproduction? It ensures the continuity of life on the earth, and it is also essential for the survival of new species. Right? Now we'll see the different types of reproduction. There are two different types of reproductions, sexual reproduction and asexual reproduction. Sexual means the production of new organisms by use of their gametes or the use of their sex cells. This is called as sexual reproduction. It is seen in the case of animals and humans. Asexual reproduction is the production of new organism from a single parent. If only a single parent is involved without the use of any gamete, then it is called as asexual reproduction. So here examples are mostly the unicellular organisms like amoeba and some of the multicellular organisms like hydra. Now, the frequently asked question in board exams is the difference between sexual and asexual reproduction. So, what are the differences? Sexual means it involves the fusion of male gamete and the female gamete. While in the case of asexual reproduction, it does not involve the fusion of male and female gamete. In the case of sexual reproduction, two parents are involved. In the case of asexual, only one individual is involved. Here, the sexual reproduction, there are chances of variation. So, the offsprings may look different from the individuals or the parents, but in the case of asexual reproduction, individuals produced are exactly similar to the parent, they are clones. It is a slow process while it is a fast process. Okay, and one more point is most animals reproduce sexually, but most of the organisms are reproducing sexually, and few organisms are there which follow the asexual mode of reproduction, which are having a simple organization like algae and fungi. So this 5 points are important, which can be asked as a difference question. Next, going to the types of asexual reproduction, there are different types of asexual reproduction which is seen. The first one is fission. Fission means, as the name suggests, it means to split or to divide. When an organism divides to form the another organism of its own kind is called as fission. So this type of reproduction is fission. There are two types of fission, binary fission and multiple fission. In binary, two organisms are produced. You can see from a single amoeba, it will extend and break into two to form two daughter amoebas. So it is called as binary fission. Bi means two, single organism splitting into two. So first the cytoplasm is dividing and then the nucleus will divide into two daughter amoebas. This is also seen in the case of leishmania, which divides longitudinally. So here it is not a longitudinal division, it can be in any plane. In the case of amoeba, it can divide in any plane, while in the case of planaria, leishmania, it will divide only in the case of longitudinal. It is dividing longitudinally, and it depends on the type of it depends where the tentacles are present. There it will divide into two. So the whip-like hair-like structure which is coming out on the same orientation, it is splitting into two leishmanias. So it is a longitudinal division, and here there is no proper orientation of division in amoeba. Leishmania, both are important and the difference can be asked based on the orientation of division. Second type of division is multiple fission. Multiple fission means it is seen in the case of plasmodium. In the case of plasmodium, what happens? The nucleus is dividing to form multiple nuclei inside the cyst. This thick part is called as cyst, inside which the nucleus keep on dividing and multiple plasmodiums are formed. And now when the cyst breaks open, it will come out and forms multiple plasmodium. So this is a multiple division where a single organisms give rise to multiple organisms of its own kind. Second type of asexual reproduction is fragmentation. The word fragmentation means breaking. You know, fragments means to break. An organism breaks into multiple fragments. But when it will break only when the parent organism matures. When the parent organism mature, it will break down into separate parts, and each fragment will grow into an individual organism. So this type of fragmentation is seen in the case of Spirogyra. It's a kind of an algae. So you can see here the single fragment is breaking into two multiple daughter Spirogyras. So this is the second asexual reproduction, fragmentation. Moving to the third one, the third type of reproduction. Before going to the third type of asexual reproduction, let's see the difference between fission and fragmentation, which is also usually asked. So fission always takes place in unicellular organism while fragmentation takes place in the multicellular organism. This is a major difference between two types of asexual reproduction. Next is regeneration. Regeneration is the process of getting back the whole organism from any of its body part. So this is a diagram given in NCERT of planaria, where you can see the planaria has been divided into three parts. Now each part is growing into the full organism. So this is called as regeneration. A single body part can regenerate into the whole organism. Clear? So now moving to the next type, that is budding. Budding is a very simple type of asexual reproduction in which a new organism is developed from a bud. So as the word suggests here, bud plays a very important role. From the existing organism, a small bud will develop. This is a bud. From the hydra, it is coming out. Slowly, it is growing to form small tentacles, and later on, it will grow and get detached to reproduce and develop into an individual hydra. Okay, so new individual daughter hydra is being formed from the parent. So this is also a type of asexual reproduction, that is budding. Moving to the next type, that is spore formation. Mostly spore formation questions are asked, which is very important, and you have to learn the diagram also. In spore formation, what happens? The parent produces hundreds of microscopic reproductive units called spores. So you can see the structure of rhizopus, which is an example. Rhizopus, which is also called as a bread mold. How this bread mold will form: when the bread is kept in the moist conditions, the spores will grow on it. So this is a like this rhizopus will grow on it. This rhizopus, how it will grow? It will have a branch structure which is called as hyphae, and on this tube-like structures, small blob develops which is called as sporangia. Sporangia contains multiple spores. So when the conditions are favorable, this will burst open and all the spores will come out and grows into and will spread throughout and leads to spore formation. So when the spore case bursts open, it will spread all over, and it is a kind of it is a airborne spore which can land anywhere, and it can grow and germinate into multiple spores and form the new spores. So this is a type of asexual reproduction where spores are playing a very important role. So they germinate faster and they are light in weight and can be easily carried away by the air. Next type of reproduction is the last and the final type is vegetative propagation. Vegetative propagation is the production of new plants which are not obtained from the seeds, but they are obtained from the vegetative parts like stems, roots, and leaves. So we are, without the help of any reproductive organs, we are developing the new plant. So there are two types of vegetative propagation: natural and artificial method. In natural method, like in the seen in the case of bryophyllum at the notch leaf margins, or at this notch of the leaf, you can find margins, you can find the small buds are developed. Okay, so the small buds, when get detached, it will fall down and it will grow into the individual or a new leaf. So this is a kind of natural vegetative propagation. The second type is artificial, which is seen in the case of sugarcane, rose, and jasmine. They have non-viable seeds. So in that case, vegetative propagation plays a very important role. So they can be produced by cutting, layering, and grafting. These are the three types of artificial vegetative propagative methods. So let's see what is cutting. In the case of cutting, as the name suggests, any small part of the stem is being taken, it's cut out, and it will grow into a new plant. It can be seen in the case of common money plants and rose and all, we go for cutting. So cutting a piece of a plant and producing a new plant is the common method which is an artificial vegetative propagation method. The second one is layering, which is shown here. The second method, the first one is cutting, the second one is layering. Layering, what happens, it is normally seen in the case of plants like jasmine, where we bend down the branch down and cover the branch with the soil. So after a few days, the roots will start developing and a new plant will develop. So this is known as layering method. So layering means bending the flexible part of the stem into the soil, covering it and trying it to grow into a new plant. The third and the last type of artificial reproduction is grafting. Grafting means we are taking two parts of the plant, like the upper part is called as sign and the lower part is called as stalk. So we are sealing both the parts and letting it to grow into a new plant. So why we need it? We have to grow a new plant with new qualities. If we have two different types of plants with two different properties, we want the both the properties to be present in the same. Now let's see the last type of a propagation method, that is by the method of tissue culture. So in this method, we are taking a small piece of tissue of any of the plant and it is placed in the artificial medium. It can be agar gel too. So when it is placed in the artificial medium, it will grow into a group of cell or a mass of cell which is called as callus. Now callus is further transferred into a medium which is containing hormones which grows into a plantlet. Plantlet can be further placed in the soil and it will grow into a new plant. So this complete first part of the process is carried out in the laboratory under very sterile conditions. So this is also a very good method for asexual reproduction. So here we have completed the first part of reproduction and see you soon in the next class with the sexual reproduction. If you like it, please subscribe to my channel. See you soon. Bye.